I'm Dr. Layne Myhre. I'm a researcher at KPU. This is going to be in three parts. I'm going to talk a little bit about the background, which I'm sure most of you already know quite a bit about, but I just want to sort of put the context of why we're doing this study out there. Then I'm going to talk about the toxicology side of the project, and then I'm going to finish up with some of the really exciting stuff that we're doing with environmental DNA monitoring. And the basic premise here is to monitor general health of the watershed. And I'll explain more about what I mean by that as we move along. And we're doing all of this in partnership with the Coquitlam First Nation. So they've been working very closely with us in this project. So just a little bit of background preamble. And I'm sure most of you have heard talks about some of these matters before. As it was said, the Coquitlam First Nation traditional territory does include all of the Coquitlam watershed. And so the watershed and the salmon that dwell in that watershed have always been of extremely great cultural importance to the Coquitlam First Nation. And so that's part of why we reached out to the First Nation in the first place to put this pilot project together. And I'm sure you've all heard about the Coquitlam sockeye, the specific subspecies of sockeye that is quite threatened and has been an area of really interesting research. Again, I'm not going to go too much into this detail, partly because this isn't really my story to tell, but also because I know you've all heard a lot of this. You're all familiar with the story of this Coquitlam sockeye, that the building of the Coquitlam Dam in the early 20th century cut off the lake from the lower watershed. And that process led to the total loss of the salmon run. There was a spring salmon run that was very culturally important to the Coquitlam people. And that salmon run was completely lost. The salmon were no longer in the lower watershed. And as you all know, relatively recently, it was discovered that there were survivors that were trapped in the upper watershed in the lake. They had lost their anadromous life cycle, meaning they didn't go back to the salt water. They were trapped entirely freshwater as effectively kokanee for their entire lifetimes. And really, really exciting work is now being done to trap these fish and to release them in the lower watershed and try to reintroduce them. And of course, that leads to the hatchery project and the building of the hatchery. I've marked the rough location of the hatchery project on this map just to show the localization here. The city of Coquitlam is marked in red on the map, and you can see the divide between the lower and the upper watershed. And I'll explain what the stars on this map are a little later, but you can see the extent of the watershed, all the various tributary streams that come into the watershed. So that's the preamble. We're very interested in the health of this watershed for a wide variety of reasons. Obviously, that affects the humans that live here, but it also is very significant to the Coquitlam First Nation. And of course, we're very interested in what's going on with the salmon that live in this river and the salmon that we're trying to reintroduce to this river. So I'm going to go into the what we're actually doing, and I'll start with the toxicology side of things. And the idea here is that we don't do a lot in general of water monitoring. When we do toxicology, we tend to look for specific events that cause specific problems. And that means that we often are caught off guard by what's really going on. And the key problem of modern toxicology really boils down to the fact that industry can come up with new chemicals that go into our watersheds far faster than we have the ability to study them. I have a slide I didn't put in here, but from when I teach toxicology, showing the exponential growth in various sectors in pharmaceuticals, in pesticide, in synthetic household chemicals. All of these are exponentially growing sectors, whereas the research publication rate on synthetic chemicals has been about the same for the last 25 to 30 years, which just tells you, we can't study this. We can't study this rapidly enough to determine emerging pollutions of interest. Not until it's already out there and it's already causing problems. So human activity impacts water systems in a lot of different ways. Some of these many people are familiar with. We hear a lot these days about microplastics from breakdown of plastic wastes. People have been hearing for years about hospital wastes in landfills causing toxic leaching, agricultural runoff from fertilizers and pesticides. Some things that people think of a little bit less include household chemical use. We think of these as things that exclusively go into wastewater and therefore go through treatment, but most household chemicals do not get removed by water treatment procedures, because water treatment is primarily geared towards destroying living pathogens and not removing chemicals. Not only that, but we don't think about the household chemicals that we put on our bodies and then we go out in the world and shed into the environment that doesn't go into wastewater. So for example, people put lotions and skin creams and medications and sunblock on their bodies. They go for a swim in a river. All of that goes directly into the water. There's no wastewater stage. We also don't think so much about things like motor vehicle breakdown. There's some really good research recently in the effects of motor vehicle tire rubber that wears down over time and the impacts that that has on salmon populations. Things like construction and mining, any kind of earth disturbance, even if it's just a construction dig, takes things that have been sequestered below surface level and exposes them to the surface, which means that new rainfall can dissolve compounds that weren't previously exposed. And those are chemicals that we don't even necessarily know what they are. And then overall, for all of this, the big thing that we don't have the ability to study well is how these interact. Because we may learn a lot about a specific chemical that we know is toxic and is causing problems, and then we can do mitigation efforts, but we don't necessarily know these are all chemicals. They interact, they do chemistry with each other. And so we don't know if one site that's producing one chemical at a safe level and another site that's producing a different chemical at a safe level, what happens when those two chemicals get together. And so this is a major problem in toxicology and in watershed health monitoring. And there are some newer strategies to try and mitigate these problems and figure out how we can study this. And I'm going to talk a bit about some of those strategies that we're trying to implement with our pilot study. Now, it's not about the tools. We have a lot of good tools for studying toxicity. One of the most common of these are bioassays with living organisms. The fish embryo test, for example, is used worldwide. It's well regulated by the Organization for Economic Cooperation and Development. And the basics of the fish embryo test are you take small laboratory fishes, you collect eggs or embryos from those fish, and then you use those in very controlled circumstances to expose them to chemicals of different concentrations. And with this, we can do things like measure all kinds of toxicity, including neurotoxicity and specific organ toxicity. The easiest thing to measure is just acute toxicity, which means the mortality, the lethality of the chemical on the fish embryos. And this is used to calculate a metric that we call the LC50, the concentration of 50% lethality. And that number is used worldwide by government regulatory bodies everywhere to determine safe usage. So you'll have a number that's set below an LC50 that is particularly low is very toxic, and a chemical that has a very low LC50 might be regulated so it can't be used in a context that has any potential to get into the environment. Or if it's above a certain level, it might be safe to use in small quantities. And this is all down to the individual governments and their regulatory groups. If that sounds like a kind of very sketchy way to decide whether a chemical is safe or not, that's because it is, but we've been using it now for decades. And it's, in many cases, the best thing we've got. But again, you have to know the chemical you're studying in order to do these tests. You have to actually take a specific chemical, look at it in different concentrations in order to get an LC50. And I'm talking about this because it's relevant to what we're actually doing. So anywhere where a watershed has a high urban impact from human activity, you can see the collection of pollutants. You can see on the left here, this is from a study in China where they were looking at relative pollutant concentrations, and they built a heat map along the watershed, and you can see as we go from blue to red, the concentration just goes up and up and up as more and more tributaries are bringing more and more human activity or chemicals resulting from human activity together. And so this is why we really want to monitor a watershed so that we can see what's going on generally. And we often think about the two major control points here, wastewater treatment and waterworks, where we treat drinking water. And of course, the drinking water in Coquitlam is not a concern because we get the drinking water only at the high end at the upper watershed away from urban activities. But that doesn't protect the impacts on wildlife, right? Because they don't get their drinking water only from the reservoir, they're getting it from within the urban areas. And of course, satellite images show us the development of the Coquitlam watershed is quite heavy. The lower watershed is over 80% developed with urban habitation. And just to bring back the previous map where you can see just to divide the upper and lower watershed, you can see the outline of the Coquitlam city boundary. And then in the lower right is Port Coquitlam. So obviously all of this has pretty significant impact on that lower watershed. So we expect to see pollutants being collected here. And the question becomes, how do we measure them? How do we see what's going on? And so these new strategies that have been coming around, particularly in Asia and Europe, are basically focused on looking at the total toxicology, the total toxic effect of the water directly from the watershed. So instead of looking for how much of chemical A is present, we know that lead is toxic. We know that cadmium is toxic. We can control those things by controlling activity that produces them. We can test the water to see if those heavy metals are present. But that doesn't tell us everything that's going on in the water. It doesn't tell us what we don't know, right? Which is all these new emerging chemicals that may or may not have toxic effects. So the idea is to take water samples from the watershed and to look at the toxicity of the water itself by concentrating that water many, many times and seeing what impacts it has on bioassays like the fish embryo test. So we're basing our methods on this large multi-governmental study that was done in Europe, the Danube River study. This was published in 2019, or one of the results of this study was published in 2019. And the basic idea was they took samples from just regular sites that they numbered all along the Danube River. They took those water samples and concentrated them many times. And then they did a series of bioassays. They did quite a few. They looked at things like estrogenic effects, so hormone-based toxicity. They looked at neurotoxicity. They looked at genotoxicity. So in other words, the ability to mutate DNA. And what they saw, and this is just the graphical abstract from that paper, so I won't go too much into this, but what they saw was given sites, and I've got some highlighted boxes at the bottom, that had more toxic effects than others. And this allowed them to identify areas along the river where human activity could be contributing in ways that we didn't know to toxicity. Just pulling out one specific figure, and this is very much what we're planning to reproduce. This figure shows just a selection of the numbered sites along the bottom from 8 to 67. And this is an LC50 test, a fish embryo test looking at lethality. You can see there's a lot of variability. They're concentrating the water hundreds of times. But you can see, and I should have, oh, maybe I don't. I'm just looking for my laser pointer function. Everybody see that? Perfect. And so you can see certain sites, particularly this one here, number 32, where you have a huge increase in the toxicity because the number of times that it's been concentrated is much less to give you a 50% lethal effect. And this was actually at the site near the city of Novi Sad in Serbia. And this led to mitigation efforts as they realized that this is a site of particular concern. They then took samples from those sites and they fractionate them to separate the compounds into different groups, usually just based on the mass. And you could actually find a given fraction, in this case, fraction 4, which is just an arbitrary number, that had by far the highest toxic effect. compared to a known toxin control on the side here. Same thing when they looked at sublethal effects in the fish, and you can see on the left some examples of sublethal toxicity, some unusual development that's not looking normal. Again, they saw this in the same fractions. They looked at failure to hatch. They looked at other things that aren't on here, genotoxicity. This EROD activity is estrogen hormone activity. And so the point is that this allowed them to actually determine not only which site along the river had the most toxic effects, even without knowing anything about what was in that water. They were then able to find fractions of compounds that were responsible for the effects and then start working to identify the compounds that were present. And even before you identify the compounds, you can start a mitigation strategy to start doing things like biochar remediation, or look at the industry that's going on in that area to look for what might be pumping into the river. So this is the kind of strategy that needs to be adopted going forward, this kind of general relative toxicity of the water itself in different regions. And this is what our pilot project, one of the things our pilot project is attempting to do. So at Coquitlam University, we have an environmental protection technology program where the senior students have to do co-op based training, and they go out during their senior year and they do site sampling and do toxicological analysis on it. So we've partnered with them to have the students in the senior program go out with water guardians provided by the Coquitlam First Nation to guide to the sites. And then they can go and they're doing our water sampling. We've already done a bunch of preliminary water sampling in November. We're going to do some more starting in February. For the time being, because it's a pilot project, we've chosen only 5 sites along the main watershed. And I've marked those here with the yellow arrows. This is also what the stars on the other map was representing, were our sample sites. And you can see what we have: one sample site upstream of most urban activity. This is near the substation. And this is our as close as we have to a control site. Ideally, we would eventually like to be able to take water from the upper watershed before any human activity as a baseline. But obviously that has a lot of special problems getting there, for example. We have 2 sites sort of in the industrial zone, one right within some of the mining activities at one of the major tributaries, and one just below all of that at the beginning of the urban residential areas. Then we have a site right in downtown Port Coquitlam underneath or after the traffic and footbridges. And finally, we have a site near the outflow. Now, as a pilot project, this is not going to give us a lot of information, but we expect to see significant differences. And even with our preliminary water sampling, we've already seen some major interesting things. The first water samples we took from Site 4 in downtown Port Coquitlam, when we just did our initial test metrics, which are things like pH and water salinity and hardness. We actually found a pH of almost 10, a very alkali water sample. And what was very interesting about that to us is the next sample we did 1 week later was back down to a normal range closer to 8. So what that tells us is that there is something that was going on temporarily that was impacting that water. And that's the sort of thing that may be connected to, city maintenance schedules. It could be related to something that was being done to treat, pests in trees along the river, or it could be something to do with road maintenance. It could just be that there was nice weather and a lot of people were in the river, in the dog park and playing in the river and bringing their lotions and sunscreens and things like that. So this is exactly the sort of thing we're trying to do with this water monitoring, is to start building this data set where you can start looking at, well, what's going on at that site at that time that caused that problem. And so when we get we're still working on our drying protocols, and when we get our concentration on that particular sample, we're hoping to see what kind of differences in toxicity that actually results in. So I'll leave that there for the first part, the toxicology, and now I'll talk about the last part of my talk here is the eDNA monitoring that we're doing. And this is very exciting because it's very cutting edge. Currently, eDNA, which stands for environmental DNA, is becoming a really, really powerful tool for monitoring biodiversity and measuring biodiversity. And this is another really good metric of a watershed health, because it's one thing to look at toxic effects in a bioassay on something controlled like a fish embryo. But knowing what's going on locally to things like the microbe species and the fish species that live in that water is obviously more interesting to us in terms of looking at the output of that toxicology, right, of the toxicity in the water. And every area in the environment really has environmental DNA. The idea here is that because DNA is so stable, which is why CSI works the way it does. They find DNA at crime scenes and can use it to identify suspects and things like that. Any place where you have organisms living means you have individual organisms that die and decompose in that area. That sheds DNA into the environment. You have spawning going on, which releases gametes, eggs and sperm. That leaves DNA residue in the environment. Any organism that is eating in an environment is defecating, and that leaves gut cells behind. And then just normal shed of scales and skin cells. All of this provides DNA into an environment. And when you look at the DNA that you collect in an environment like a watershed, you will see traces of all the species that are contributing to that DNA. And you can do bioinformatics with computational tools to determine relative levels. So you can see, for example, if a given species is very threatened or less threatened in an area based on its relative abundance of environmental DNA. You can see whether invasive species are coming in because you'll see increasing amounts of environmental DNA and so forth. So we're in the pilot project, we're focusing on microbes and bacterial species because that should give us a very interesting metric for what's going on with human activity and impacts. But we're also interested in looking at the salmon themselves and looking for their eDNA. Now, this requires a lot of very powerful tools. This is the basic workflow for an eDNA project. You have your sample collection, which we're already doing for toxicology testing. You spend a lot of time filtering all that water down into a microfilter to capture all the DNA that's present. And then you process that DNA. And then you need to do genomics and bioinformatics. And this requires powerful tools. This is the hard part, actually, doing the analysis, sequencing everything that's present, and then using computers to pull out the information. And luckily at Coquitlam University, we have an institute called the Applied Genomics Center. It's only been around for a few years now, but they already have, I think it's going on 30 different industrial partners. They mostly are working in the agri-tech sector and doing agricultural genomics, but they're also doing now more and more work with environmental DNA measurements. And so that means that we have the tools for this kind of sequencing and for the bioinformatics. So in the pilot, we're just going to look at all the bacterial species that are present and start looking for species of interest. On one hand, we just want to see changes between the different sites. But on the other hand, we're interested in things like emerging pathogens, particularly pathogens and bacterial species relevant to the salmon. Where salmon populations are healthy, we expect certain bacterial species that have co-evolved with salmon, that live with salmon, that are gut bacteria for salmon to be present. Where there may be threats to salmon, we expect to see pathogens. So this is something that we're going to be taking those water samples from the same 5 sites and looking at. We also are going to be looking at the salmon DNA itself. We want to determine how accurately we can measure salmon DNA levels. And using this, we're hoping to develop more exciting tools going forward. We have actually put a grant together and submitted to the Fish and Wildlife Compensation Program to expand both the toxicology and the eDNA aspects of our pilot project. And this is actually a figure from the grant that we put in about the workflow. And the idea would be that if we get the funding to greatly increase our sites where we're doing sample collection, we should be able to take DNA from and now I'm back to the preamble at the beginning, the Coquitlam sockeye that we're very interested in the health of. We should be able to take the DNA of those organisms and sequence it and do comparative genomics with the very well-established sockeye salmon genomes that have been published, that are very well annotated. And that means we can look for areas of variability so that we can actually trace individual lineages and see, are salmon that are being released from the hatchery program actually surviving in the watershed? Where are they surviving in the watershed? Are they returning to spawn? Where are they spawning? All of that without having to be able to trap and pull out individual fish and take tissue samples and do other time-consuming invasive work. We can just look at the water samples and the eDNA that's present. And this allows to build things like this heat map. This is just an example heat map where you can see areas that are red are the densest or highest relative component of a given species. So if you imagine looking at the watershed map like this one, if we reach a point where we have enough sampling that we're looking at every branch, every major tributary branch along the watershed, that will tell us what species are present upstream of that branch point. And this is going to be a really powerful tool for assessing the success of the hatchery project and other restoration efforts. And this kind of monitoring ongoing, we're hoping we can use to build models to apply to other watersheds, looking at the impacts of human activity on the ecosystem, the wildlife that are present through eDNA monitoring, and looking at the toxicology through and then, of course, putting this all together, when we start looking at these maps of where there's toxicology happening and looking at what the species are present, both in microbes and in macro species like salmon, we can start to look for correlations. And we say, oh, the salmon are coming back, but they're only thriving in this area, not this area. Why? Oh, we're finding toxic effects in the water in this area. Maybe that's what's impacting the salmon there. And that helps us to target any sort of mitigation efforts. So that's the big picture, what we're hoping to get to eventually with our little 5-site pilot project as we expand. And we've already had interest from other funding agencies. I've spoken with people who are involved in the BC salmon restoration program, and Genome BC is very interested in eDNA monitoring programs. So we're hoping that we can get the funding to expand this.